Section 1.4 in your book is fairly brief, but I think that cellular structure is important to the understanding of biochemistry, and I'm consequently going to spend more time on it. The first thing to point out about the cell is arguably the most important part of the cell, and that is the cell membrane. I'm showing here a small snippet of an electron micrograph of a plasma membrane. I want you to notice that I'm interchangeably calling this the cell membrane and the plasma membrane. And I would really encourage you to take a brief look at electron micrographs of all of the organelles that we're going to talk about. In my on-campus course slides, I have electron micrographs which I have collected from various places, but copyright makes it impossible for me to share them with you in an online course. If you're interested in electron micrographs, I would encourage you to visit Dr. Jastrow, that's Jastrow page at the University of Mainz in Germany. He has an outstanding collection of images for which the only problem I think you may run into is that the text on many of his pages is in German. But as this image shows a plasma membrane, it appears as 3 layers by electron microscopy. The upper left portion is the outside of the cell, the lower right portion is the inside of the cell, and the dark, light, dark stripes are the plasma membrane. Another thing to note about the cell membrane is that its thickness varies with time. I'm giving here a range of 7.5 to 10 nanometers thick, which is on the heavy side. I probably should say like 5 to 10, but the thickness varies in size because it is composed of fatty acids, and those fatty acids change during the course of a year. And we'll talk about that later in the course. The 3 layers, which is composed of the fatty acids, that's 5, 7.5, 10 nanometers thick, is shown schematically in figure 1.9 of your book at the bottom left of the slide. The reason that the cell membrane is so important is that it functions as a barrier between the living and the non-living. All cells, whether they're prokaryotic, eukaryotic, or archaea, have a plasma membrane, and we will explore membrane structure in much more detail later in the course. The next organelle I want to highlight is the nucleus, which is arguably the most important organelle of the cell. I have a small image of an entire cell which has been stained and examined by a fluorescence microscope. Although fluorescence microscopy is not a workhorse tool in every biochemistry lab, this picture demonstrates the power of fluorescence microscopy for looking at multiple structures relative to each other. The nucleus of the cell in this image is black, as I did not stain it. And I wanted to show this image to show that the nucleus occupies a large portion of the cell. The bottom part of the slide, which is figure 1.12 part A of your text, shows a schematic diagram of the entire cell where the nucleus is labeled in purple, and at the right of the electron micrograph is an electron micrograph of the entire cell. And again, what you can see is the nucleus occupies a very large portion of the total area of the cell. So 3 points. The nucleus occupies a very large portion of the cell. 2, the nucleus often contains 1 to 4 nucleoli. In this figure, there's only 1 present. Third, though not very evident in this electron micrograph, is that the nuclear membrane is actually very porous. This will be important late in the course. Next on my list of cell structures is the cell wall, which is not present in all cells, but rather only in prokaryotes, which, by the way, will not contain organelles, and plants, which will contain organelles. The cell wall is not much to look at, but it is thick and tough and has a function of maintaining rigidity. Plants can survive both inundations and droughts because of the cell wall. The next organelle is the mitochondria, which is arguably the most important organelle of the cell. As I would show you in person with an electron micrograph, the size and shape of mitochondria varies enormously. This variation depends on the type and shape of the cell. In addition, mitochondria divide by binary fission and thus necessarily would be 1/2 as large after division. Binary fission is one of the criteria used to support the claim that mitochondria arose via endosymbiosis. One last point about mitochondria is that although there is 1 plasma membrane and 1 nucleus and 1 cell wall, there are many mitochondria per cell, again, depending on the cell type. A cell which is very metabolically active will have a larger number of mitochondria than, say, an adipocyte, whose function is only to store fat. We will examine mitochondrial structure when we are discussing metabolism, but for now let me point out the several compartments of the mitochondria: the outer membrane, the inner membrane, the matrix, the intermembrane space, and the cristae. The function of the mitochondria is the powerhouse of the cell. All eukaryotic cells, whether it's plant or animal or yeast, are going to have mitochondria. There are the structures, the compartments, and we're going to talk again about the structure and compartments when we get to the metabolism section. The next cellular structure I'd like to discuss is the endoplasmic reticulum, which is arguably the most important cellular structure and is composed of 2 types of ER, rough and smooth. The endoplasmic reticulum is a perinuclear phospholipid-bound structure, phospholipid-bound as all organelles are. If bound by ribosomes, it is referred to as rough ER, and if rough ribosomes are not present, it would be referred to as smooth endoplasmic reticulum. These 2 types of ER have different functions, namely that rough ER functions in protein synthesis and smooth endoplasmic reticulum is involved in lipid synthesis and also in calcium metabolism. Referring back to figure 1.12 part A, you will notice a great deal of endoplasmic reticulum in that cell. It is a blue structure in the schematic diagram, and the tube-like structures in the electron micrograph. The Golgi apparatus is a sac-like structure located further away from the nucleus. It should also be noticed that it is composed of numerous sac-like structures, which are referred to as the cis-Golgi, the medial Golgi, the trans-Golgi, and the trans-Golgi network. Golgi bodies function in protein processing and secretion, and secretory cells will have more Golgi bodies. You might notice here that I stopped my running humor about being the most important organelle. It is my hope that it becomes apparent that all of these organelles are highly important to the cell. Remember, everything has a function in nature, or nature would not have kept it. Lysosomes are an organelle which is present in all animal cells, but not in plant cells. They're approximately the same size as a mitochondria, which again I would show you with an electron micrograph if you were on campus, and they function in protein degradation. When we look at them under the electron micrograph, it looks like just a black blob. So because they function in degradation of proteins, the number of lysosomes in a cell will vary considerably. I should also point out that plant cells contain a similar functioning organelle referred to as the peroxisome. The lysosome uses enzymes to degrade proteins, whereas the peroxisome uses hydrogen peroxides. Next on my list of organelles is chloroplast, which is an organelle present in plant cells, but not in animal cells. Like mitochondria, this organelle is thought to have arisen by endosymbiosis, and like mitochondria, it contains internal membranes referred to as thylakoid discs, which always occur in stacks called grana. The function of chloroplasts is photosynthesis and is therefore present in all photosynthetic eukaryotic cells. The number of chloroplasts varies by plant, but because chloroplasts are larger than mitochondria, there is perhaps up to about 100 chloroplasts per cell. Bathing all of these organelles is the cytoplasm. Notice that in some biology books the term cytosol is used, but these terms should not be used interchangeably. Cytoplasm is composed of cytosol and organelles. Cytosol is obtained by breaking open cells and centrifuging the broken cells at a very high speed for a very long time, 100,000 times G for 24 hours in the parlance of biochemistry. This results in a precipitate of organelles and the supernatant fluid known as the cytosol. The organelles have in common that they are all membrane-bound structures, as I said above a couple of times. We will now turn our attention to important non-membrane-bound structures. The first of these are the microtubules and microfilaments, which are composed of the proteins tubulin and actin, respectively. Though not organelles, these are very important for giving the shape to the cell, as well as functioning in transport. I next want to point out that when you consider a cell having as much volume taken up by the nucleus, which in turn is stuffed with DNA, And that cell is also containing mitochondria, tubules, Golgi bodies, microfilaments, endoplasmic reticulum, lysosomes, etc. The cell has to be a very crowded place. Cell biologists like myself believe that if you could palm a cell like a basketball and use a knife to cut off the bottom of the cell, it would not be like popping a water balloon where everything gushes out immediately, but rather we believe that the cell's contents would slowly ooze out.